Thank you. I'm glad to welcome everyone today. So let me introduce our research analysis of environmental adaptation features of the bacteria of the Flavobacterium genus by comparative genomics. Bacal sponges of the species Lubomirskiia bacalensis are the object of our interest, and they are the habitat of symbiotic organisms like bacteria and green algae. For many years, we observed disease development in these species. A shift in the composition of the microbiome in the metagenome was found in diseased sponges. There is a hypothesis that this is the reason for their damage. One strain of the bacteria whose composition has significantly increased in diseased samples is Flavobacterium SLB02. It became the subject of research in this work. Bacterial phylogeny based on 16S rRNA sequence was performed using sequences from GenBank. And although our strain turned out to be close to the species of Flavobacterium hidatus, a different picture was observed after adding sequences from metagenomic assemblies. The phylogeny based on 16S rRNA gene didn't let us to classify the desired strain to any species. Tree turned out to be unstable and bootstrap support was low. Although to check whether the species inhabited the aquatic environment, we took samples from the water column and sediments, collected sequences encoding 16S rRNA and built a tree, but the results also didn't reveal close species. There is a possibility that the strain got into the aquatic environment by accident and it's not widely distributed there. But the phylogeny based on the whole genome showed that the desired strain is close to the terrestrial and defined strain of leaf 82. Despite the fact the strain was found in an aquatic environment, it has some features, characteristics of terrestrial strains. For example, they are the size of the genome and the number of genes. And we are interested in whether this strain has adapted to a new habitat, what properties contributed to this adaptation, what features the organism has acquired. This task can be expanded to the task of determining the features of adaptation to an ecological niche. Representatives of the Flavobacterium genus are characterized by the ability to decompose a wide variety of polysaccharides. Initially, a comparative analysis of the metabolism of polysaccharides of about 50 strains was carried out. The dbCAN tool was used to detect polysaccharide utilization lots, abbreviated as POOL. As a result, a comparative table was obtained for the analyzed objects. It reflected the quantitative distribution of genes encoding enzymes for the decomposition of polysaccharides by enzyme classes. The multi-feature space was mapped on the plane using principal component analysis. This work made it possible to compare the studied strains visually by the ability to utilize polysaccharides. Initially, the strains were labeled as terrestrial and aquatic ones. We managed to group the studied samples in some way. The blue markers are conditionally water samples, and the red markers are terrestrial ones. It can be assumed that according to the genes encoding the enzymes of polysaccharide metabolism, it is possible to attribute the strain to one of ecological niche. In addition to the utilization of polysaccharides, other subsystems are also interesting for comparison between different strains. We have chosen a service for annotating prokaryotic genomes of Rust and its hierarchy of biological systems. An additional tool was needed to process and analyze the annotation results. Such tool was developed during the research. It makes it possible to classify the results of functions annotation using Rust classification and keywords classification. Thus, data with unclassified functions is received at the input. As a result, a table with classified functions is formed. There are functions that are not classified by Rust, and if necessary, some of them can be classified by keywords. The classification results are then used as input data or for calculations. Calculations depend on the biological parameters selected by the user. Their hierarchy is taken from the Rust system. The result is a table where each strain has a multidimensional vector, where each number reflects the number of genes within the selected parameter. Since the calculation results can be multidimensional, it is necessary to reduce the dimension for the visualization in two-dimensional space. There are some methods for this. We took PCA, MDS, and TSNE for our application. All three methods generally give different results, but the topology of the distribution of strains is similar for all methods. This software product made it possible to obtain visual data on which strains that are genetically similar in some way located close. During the analysis, it was possible to make a comparative analysis for the studied samples. For example, the quantitative distribution of genes responsible for the metabolism of factors, vitamins, pigments, and prosthetic groups is similar for all strains. But some features have also been identified, and the difference between which values in terrestrial and aquatic strains are quite distinguishable. They are the metabolism of nucleic acid, including the CRISPR system, and the metabolism of hydrocarbons, mono, D, and oligosaccharides. The results of the separation on these groups gave good results, which allow us to divide the samples into two ecological groups. There are some strains that are out of the picture. We assume that this is due to several reasons. There is a possibility that functions that the Rust system didn't identify may affect the result after taking them into account. Some functions may have been incorrectly classified or unclassified at all. In addition to the imperfection of Rust system, errors could occur at the labeling stage, since not all habitats of strains included in the analysis are reliably known. The question of dividing strains into ecological niches remains open. Is it worth isolating strains found in water environment, in glaciers or polar reservoirs from the aquatic clade, or is it worth dividing the terrestrial clade into endophytic and soil strains? As a result, for additional labeling, we tried dividing the results into more niches. The results showed that endophytic strains stand out noticeably. Also, the strains found in sediments from a small group based on these characteristics. But in order to detect feature that would make it possible to isolate polar strains or soil strains into a separate group, it is necessary to continue the feature analysis. In the future, we will continue to analyze the features, try to modify the results of genome annotation by the Rust service, and eliminate the disadvantages of this system. It is also necessary to add statistical analysis to the developed software tool, which lets us to value the reliability of the contribution of a particular feature in the visual separation of strains. And that's the end of my presentation. Thank you for your attention, and I'm ready to answer to a question.
Any questions? You may ask them in Russian. I think that lady will understand you. Then I have a couple of questions.
Mhm.
First of all, the first one is sort of conceptual. In your header title, you mention adaptation. As I
Mhm.
understood from your presentation, that there are different sets of organisms living in different sets of environmental uh, characteristics. Uh,
Mhm.
why do you believe? To me, adaptation is certain efforts to adapt. So there is a selective power of environment and adaptive capacity of organisms. How can you estimate the role of adaptive efforts so that you call it adaptation to certain environment?
Mhm. I understood. Well, we assume that because of some functions that allow some strains to survive in different environments, um, uh, well, mm, wait a minute, I will construct my sentences. Um,
You may answer in Russian. That's perfectly okay.
Окей, хорошо, давайте попробую. В общем, мы полагаем, что у тех организмов, которые мы изучаем, гены все-таки выполняют же какие-то функции, какие-то кодируют какие-то белки, и тем самым определяют, с какими условиями тот или иной организм может справляться, в каких условиях он может выживать. Поэтому из-за какого-то количественного распределения генов мы можем судить о том, насколько организм приспособлен к тем или иным условиям.